In the interest of time, I'd like to start the seminar. Again, thank you everybody for coming to this neighborhood science seminar in conjunction with the Regeneron, and we partnered with Regeneron specifically to provide the community, and we are going to disseminate even larger, hopefully, to provide the community with state-of-the-art, cutting-edge science. So please take advantage of it. This is done for the community, and we appreciate the comments, questions, so on and so forth. So the title of the talk today is cell-derived type 1 interferon that instruct tumor monocyte functional polarization. Speaker is Dion Kwart, has a Ph.D. and a principal scientist at Regeneron in the immunology and inflammatory disease therapeutic focused area. He studies how innate and adaptive immunity system function to detect and respond, and we all are so critically waiting for this type of immune surveillance of the cancer cells, and how we, with the immune therapy, being able to detect and stop them to a certain extent. So prior to joining Regeneron, Dr. Kwart completed a PhD and a Master in Science in Neuroscience at the Rockefeller University and in Oxford, in the University of Oxford. He's originally from Toronto, but he doesn't speak French. And he received his bachelor from the University of Ottawa. Myeloid cells, in terms of the description, and I'm not going to steal his term, but that's what he told me, including monocyte and macrophage that are basically those myeloid cells. They have an classic immunosuppressive function, and they are the first line of defense that we have for optimal immunotherapy outcome in cancer, as well as in regular inflammation. Dr. Kwart will highlight his work aimed at unpacking the complex heterogeneity in monocyte in the tumor environment. He will describe a novel role of cancer cell-derived type interferon in driving immunostimulatory tumor monocyte polarization. Without further ado, please join me in welcoming Dr. Kwart.
Thank you. It's great to be here. As mentioned, I'm Dylan. I'm in the immunology group at Regeneron, and I'm going to tell you about work that I did mostly in my time as a postdoc at the postdoc program at Regeneron, and how we found that a potent immunostimulatory cytokine, type 1 interferon, can be produced by cancer cells themselves and drive a functional polarization of tumor monocytes that's associated with anti-tumor immunity. So, as I'm sure, and I hope we're all aware of, the goal of immune checkpoint blockade, like anti-PD-1 immunotherapy, as shown here, is to take advantage of the natural ability of T cells to target and ultimately kill off cancer cells. Now, something like anti-PD-1 will augment that function. And this would obviously lead to tumor cell control. Now, unfortunately, the targeting of a T cell to tumor cells does not happen in isolation, but rather in this pretty highly complex and often immunosuppressive tumor microenvironment. Now, what I mean by that is that there's a variety of cell types in this environment that secrete things or have different functions that ultimately dampen down the activity of T cells, and therefore they cannot kill off cancer cells or respond to immunotherapy like anti-PD-1 therapy. Now, myeloid cells are often the most abundant cells in the tumor, immune cells in a tumor. These include cells like monocytes and granulocytes, but also macrophages and dendritic cells. But today we're going to focus primarily on monocytes. And one of the reasons for that is that they are indeed a pretty large component of the tumor microenvironment. And they're these incredibly plastic cells capable of taking on different phenotypes that can ultimately play a big role in the tumor and anti-tumor immune response. So when people in the field typically think of monocytes, we think of how they can get into a tumor and pretty rapidly develop an immunosuppressive function. And what I mean by that, again, is they can prevent T cells from becoming activated or proliferate to then ultimately target and kill off cancer cells. However, in certain environments, monocytes can become pretty potently immunostimulatory. And it's possible that in a tumor, these immunostimulatory monocytes could actually do the opposite and activate or augment CD8 T cell functions or response to immunotherapy, ultimately leading to more tumor cell death and cancer control. So therefore, we and many others in the field are thinking, well, how can we potentially take tumors that have a lot of these immunosuppressive myeloid cells and monocytes and try to repolarize them to make those suppressive monocytes more immunostimulatory, or just reprogram the environment so that new monocytes, as they arrive, just rapidly become immunostimulatory, ultimately to drive T cell activity and tumor cell death. And we think this could be a potential clinically advantageous approach that would combine quite well with current existing immunotherapies. Now, the development of monocyte targeted therapies, I would say, is pretty hindered by really poor understanding of monocyte and myeloid cell diversity in general in tumors. So as I'm trying to demonstrate here, it's really unclear, for example, when a monocyte's in a tumor, what are the specific cues that allow for that monocyte to become either a suppressive monocyte or perhaps an immunostimulatory monocyte. So deciphering those specific cues in tumors is yet to be, I think, nicely done. And furthermore, I guess in general, it's not really well established or super clear if these immune stimulatory monocytes even exist in tumors naturally, and how we can identify them at all, if at all. So with that, to start, we wanted to take a pretty agnostic approach and get a better handle on the heterogeneity of monocytes in human tumors. So we started by looking at a lung tumor single-cell RNA sequencing data set. Lung tumors are pretty, known to be infiltrated by a lot of myeloid cells, specifically monocytes and macrophages. And when we pull out the monocytes specifically and do some clustering analysis, we can actually identify 3 different clusters of human tumor monocytes. We see this cluster C0 that appears to be a bit more immature, like monocyte subset based off of the gene expression. As well, on the other hand, we have this cluster C2, which we found is kind of more resembling a more immune suppressive or mature and differentiating monocyte. But what really stood out to us was this cluster C1. And this was marked by a number of type 1 interferon associated genes. And additionally, although not really shown here, there's a lot of other pro-inflammatory or immune stimulatory genes that kind of were signature to that cluster C1. So this kind of got us thinking maybe these interferon associated monocytes might actually be immunostimulatory or pro-inflammatory. Now, to be able to actually study these kinds of monocytes, we wanted to see if we could identify similar subsets in a mouse tumor model. Obviously, if we can do that, we can really get into the deep biology of these different subsets. So we looked at a mouse lung tumor model, this Lewis lung carcinoma model, and we purified out the monocytes from the LLC tumors, as well as blood, spleen, and bone marrow from these mice, and did single-cell RNA sequencing. And what you see here is that the tumor monocytes in general, they pretty much cluster separately from the peripheral monocytes, the blood, spleen, and bone marrow monocytes. But interestingly, we also find 3 different clusters of tumor monocytes. Again, we find in cluster C0 what look like more immature, potentially naive, newly arriving monocytes. We also find a cluster C2 that look more immune suppressive. They express genes like arginase, SPP1, things that are known to be associated with immune suppressive myeloid cells. But again, we find this cluster C1 that's pretty much marked by the expression of these type 1 interferon associated genes, as well as some other pro-inflammatory genes. I'm just highlighting CXCL10 as an example of that here.
Are these markers classified or retrofitted from the old classification M0 and 1 and M2, or it's a completely different display?
Yeah, good question. So we've tried to not really use those old classifications, as I think a lot of the field are doing, to call what we see now, and just really go by what we can characterize in the lab. So go by function, go by gene expression, and kind of call it as we see it. But in this case, if you were to take this cluster C2 signature and put it with like a classical M2 macrophage, there's a lot of overlap, and we've done that. Yeah.
And those monocytes were taken from the tumor by itself. Have you tried the bronchial lavage macrophage that we extract from?
Yeah.
Just to see whether there is a phenotypic change between the bronchial lavage onto the tumor by itself.
Yeah. So this tumor model in mice is a subcutaneous implanted syngeneic tumor model. So we don't really have the kind of orthotopic tissue to work with. Yeah. So as I was mentioning, we're kind of struck by those interferon-associated monocytes. I just want to highlight here the similarities between human and mouse that we found. As you can see, two of the most highly differentially expressed genes, ISG15, I53, these are classical interferon-associated genes, quite high in that cluster C1 in both human and mouse tumor monocytes. Now, given that, and given, as I mentioned, there's also these more pro-inflammatory genes associated with this cluster, we were curious, what is the role or function of this subset of monocytes? And to better be able to study that, we wanted to first find a tumor model that seemed to be enriched with this subset of monocytes. And to make a long story short, we ended up finding that the MC38 tumor model had monocytes that pretty much almost all looked like these interferon polarized monocytes. And I'll show you that here. Essentially, we did some single cell RNA sequencing, and what you're seeing here is essentially all of the monocytes from the tumors. And we're looking at those interferon-associated cluster-defining genes. And you can see that, for example, ISG15, you see most of those MC38 tumor monocytes highly express that gene, whereas only a subset of the LLC tumor monocytes express that. And so, as I mentioned, considering that there's also this associated expression of pro-inflammatory genes like CXCL10, CD86, MHC class 1, for example, we wanted to basically figure out whether there'd be functional differences. Would these MC38 tumor monocytes as a whole be more immunostimulatory because there's more of these interferon-associated monocytes? So to test that, we basically sorted out the monocytes from different tumor models, LLC and MC38, and ran them in a couple different assays. So I'll first talk about their suppressive capacity. Basically, we mix those monocytes with activated T cells and see how well they can actually suppress the proliferation and activation of T cells ex vivo. And we see that indeed those MC38 tumor monocytes, those interferon-associated monocytes, are significantly less suppressive than the LLC tumor monocytes. Now, on the other side of the coin, we want to see, okay, are they actually immunostimulatory? And here we're mixing monocytes with antigen-specific T cells and seeing, can they actually activate and promote the proliferation of these T cells? And in fact, the MC38 tumor monocytes are significantly better at promoting the activation and stimulation of those T cells. So indeed, it does seem like those MC38 tumor monocytes, which have a majority of these interferon associated cells, are indeed more stimulatory and less suppressive than the LLC tumor monocytes. Now, this is, though, looking at all the monocytes from a tumor. And as I mentioned before, by single-cell RNA sequencing, we found these different subsets. So we wanted to figure out a way to actually be able to identify and distinguish by surface marker expression these stimulatory interferon-associated monocytes from more immune-suppressive monocytes within the same tumor. And basically, to make a long story short, we ended up finding that there's two surface markers that, in addition to classical monocyte lineage markers in mice, we can use to differentiate these different subsets like we found by single-cell RNA sequencing. So we find that in the MC38 tumor model, when we look at the expression of CD88 and SCA1 on those tumor monocytes, most of the monocytes look like these double positive CD88, SCA1 positive monocytes, as shown here, whereas you see way fewer in the LLC tumor model. We're calling those cells monocyte 1 cells. In the LLC tumor model, on the other hand, we see some monocyte 1 cells, but predominantly these monocyte 2 cells, single positive CD88, SCA1 negative cells, as shown here. There also is a smaller proportion of these monocyte 0 cells, these double negative cells. We think these are newly arriving immature monocytes. So just to show you that these monocytes reflect what we saw from the single-cell RNA sequencing, we sorted out these major populations, the monocyte 1 and monocyte 2 cells. We did bulk RNA sequencing. And indeed, these monocyte 1 cells that came from MC38 tumors enriched for pathways and gene expression associated with responsive type 1 interferon, suggesting these really are those type 1 interferon associated monocytes, while these monocyte 2 cells enriched for pathways associated, typically more suppressive genes like pathways associated with tissue remodeling, hypoxia, angiogenesis, more tumor promoting. And lastly, just to show that these different subsets functionally are distinct, we sorted those out and did those same suppression and stimulation assays again. And in fact, again, those monocyte 1 cells, those interferon associated monocytes, significantly less suppressive than the monocyte 2 cells, and monocyte 1 cells significantly more immunostimulatory than those monocyte 2 cells. So here, I hope I've convinced you that we found these type 1 interferon associated monocytes in tumors. We're calling them monocyte 1 cells. And we think they're immunostimulatory and potentially an advantageous monocyte subset that you potentially want to amplify in a tumor environment to promote anti-tumor immunity. So given our interest in these stimulatory monocyte 1 cells, the question then became is like, how do these cells emerge? How do they become polarized? So to start, we wanted to ask, where does the polarization happen? Is it directly, can the tumor environment itself directly polarize these different monocyte subsets, including the monocyte 1 cells? So to test this, we did an adoptive transfer experiment. We took advantage of congenitally labeled animals. So we had recipient mice that had the CD45.1 allele, and we implanted into those mice tumors, either the LLC tumor or MC38 tumor, and then took CD45.2 donor monocytes and basically injected them right into the tumors and asked, what do these monocytes, how do they look a couple days later? So just to show you the monocytes before we transfer them into the recipient mice, they all look like these monocyte 0 cells. They're double negative, CD88, SCA1 negative. However, when we take these monocytes, put them into an LLC tumor, a couple days later, they pretty much all look monocyte 2-like, just like we saw from tumors that we analyzed previously. However, when we took monocytes and injected them right into the MC38 tumor, they pretty much all look monocyte 1-like, just like we saw when we analyzed tumors previously. So this tells us that the tumor environment itself is sufficient to drive this differential monocyte polarization that's pretty stereotyped and unique to these different tumor models. Now, to take it a step further, the question then became, well, is it something the tumor cells themselves are making that's driving this polarization? So to answer that question, we generated conditioned media from these tumor cell cultures, and then concentrated that media to get a nice stock of whatever factors the tumor cells might be releasing. And then we use that concentrated conditioned media to treat mouse bone marrow monocytes in vitro for a couple days. And when we have bone marrow monocytes that we treated with unconditioned media, so media that did not see any tumor cells, they pretty much stay these immature monocyte 0-like cells, double negative. However, when we have monocytes that were treated with the supernames from MC38 tumor cells, they pretty much all become monocyte 1 cells like we see in vivo. However, when we treat monocytes in vitro with the LLC supernames, they pretty much all become monocyte 2 cells, just like we see in vitro. And I really want to emphasize that these aren't just surface marker changes. These are functional changes that are happening in vitro that look like what we see in vivo. So we took these in vitro polarized monocytes and ran our T-cell assays. And you can again see that the monocytes polarized with the MC38 tumor cell supernames significantly less suppressive compared to the monocytes polarized with the LLC supernames. And flip side, when we look at the stimulation assay, the monocytes polarized with the MC38 supernames significantly more stimulatory than those polarized with the LLC supernames. So just to pause here and summarize where we are so far, basically what we've shown is that monocytes, when they arrive in the tumor, there's maybe this transition or intermediate state, this monocyte 0 cell that's a bit more immature. And then depending on the tumor, the polarization diverges. So in the LLC tumor, there's something coming from the LLC tumor cells that drive this monocyte 2 polarization. This is a suppressive monocyte that can dampen down the activity of T cells. However, in the MC38 tumor, there's something coming from the cells that is driving this monocyte 1 polarization and leading to this stimulatory phenomenon and T cell activation. So given that, as I mentioned, we're trying to figure out how to amplify this and make tumors look more like this with more immune stimulatory monocytes, we wanted to figure out what specifically is driving this functional polarization of these tumor monocyte 1 cells. So given that these monocyte 1 cells are kind of defined by the response to type 1 interferon on a molecular level, we then wanted to ask the obvious question: does response to type 1 interferon, is that required for driving this functional stimulatory monocyte polarization? So to test that, we used a mouse model that we had, which basically had the type 1 interferon receptor IFNAR humanized. And the reason why this was useful to us is because a human receptor will not respond to any mouse type 1 interferon. So it's almost like a knockout. It's not going to see any of the interferon, should that be what's driving this polarization. So we basically grew our 2 tumor lines in wild type, or those humanized IFNAR mice, analyzed the monocytes by flow cytometry and looked at their functional properties. And what we see is pretty striking. So when we look at those MC38 tumors in wild type mice, as we typically see, most of those monocytes are monocyte 1 cells, these double positive cells. However, when we implant the MC38 tumor cells in the humanized IFNAR mice, you see a significant reduction of the monocyte 1 population and a corresponding increase in that suppressive monocyte 2 population. Now, also as expected for LLC, you don't really see much of a difference in the monocyte 1 population. You don't see many of those cells to begin with, really suggesting that that monocyte 1 population depends on the response to type 1 interferon to exist. And again, just showing you that these are functionally different cells, we see that these monocytes coming from the humanized interferon receptor mice, these indeed are significantly less immunostimulatory than the monocytes coming from the wild type mice. So the response to interferon is clearly necessary to drive this immunostimulatory monocyte polarization. So this begs the question, okay, well, where is the interferon coming from in the tumor? I'd say most people in the field would think upon radiation treatment or treatment with DNA damaging agents or something like that, immune cells would likely be the main producer of type 1 interferons. But that's actually not what we found. In fact, it was quite a surprise to us that the tumor cells themselves, these MC38 tumor cells, seem to be pretty much the main producers of type 1 interferon. So this is just looking at those tumor cell cultures in vitro, those supernatants that are used to treat the monocytes. When we actually look at the levels of interferon in those supernatants, we find that the MC38 tumor cell supernatants, lots of type 1 interferon, and kind of as predicted, or this makes a lot of sense, in the LLC tumor cell culture supernatants, we see absolutely no type 1 interferon. So the tumor cells themselves are making interferon. And then we wanted to ask the question, well, in an actual tumor, who's actually making the most? So the tumor cells or potentially the non-tumor cells and immune cells. So we took MC38 tumors and actually separated out tumor cells from the immune cells or non-tumor cells, but it's mostly immune cells, and then plated those overnight and looked at the amount of interferon that was produced from those cultures. And in fact, we see that the tumor cells seem to be making way more type 1 interferon than the non-tumor cells. So this led us to really believe in this tumor model, MC38 tumor cells, on no treatments or manipulation, are just naturally producing type 1 interferons and are the predominant source. So taking it a step further, we wanted to figure out, well, why are these tumor cells just constitutively spitting out type 1 interferon? That seemed to be a little bit of a surprise to us. So we looked at the usual suspects of how type 1 interferons can be produced by cells. So there are these nucleic acid sensors pretty much in every cell to sense double-stranded DNA or double-stranded RNA, which really shouldn't be in the cytoplasm. Maybe if there's genomic damage or a viral infection, then you'll get some of these nucleic acids in the cytoplasm. And then there's these sensors to detect this and trigger basically, something bad is happening, and that leads to the production of type 1 interferons. So we basically use CRISPR-Cas9 to target different components of these pathways. And we found that when we target STING and the STING pathway, but not the double-stranded RNA sensing pathway, MAVs, we see that you completely lose the production of type 1 interferon. So the MC38 tumor cells are clearly making type 1 interferon in some sort of C-gas-sting dependent manner. So now that we know that, we ask the next question is this tumor cell-derived type 1 interferon playing a role in this monocyte 1 polarization, and specifically that functional polarization of these monocytes, or is this something else? So we took advantage of those CRISPR cell lines that I just showed you in the previous slide. We had a control cell line where the MC38 tumor cells were targeted at the safe harbor ROSA locus, so no gene expression alteration should be expected. Or we had the MC38 tumor cell line that had TBK1 knocked out, so they would produce no type 1 interferons. We grew tumors, looked at the monocyte polarization and functional activities. And what we see is that in the control MC38 ROSA tumors, again, predominantly monocyte 1 cells, but 71% of those monocytes are monocyte 1, but we do see actually the significant reduction in monocyte 1 polarization in the TBK1 knockout tumor condition. Again, we see this corresponding increase in the monocyte 2 population as well. And again, just to show you that it's not just surface marker changes, these are functionally different populations of myeloid cells. We took these ROSA control monocytes, or the TBK1 tumor monocytes, and ran our T cell stimulation assay, and the myeloid cells from these TBK1 knockout tumors that have more of these suppressive monocyte 2 cells indeed are significantly less able to stimulate T cell proliferation. So it really does seem like tumor cell derived type 1 interferon is playing a key role in this model for driving that monocyte 1 polarization and that immunostimulatory phenotype. Now, everything I showed you up until now is basically, at least the wet lab work, was all from mouse cells. So we wanted to get a handle of, does this also happen in human cells? So we looked at a variety of different human tumor cell lines. And we asked, do human tumor cells also produce type 1 interferons? And surprisingly, we found that few tumor cell lines were able to produce type 1 interferons. So we see quite a bit actually made by this cell line, NCIH1650, as well as a lot less, but some made by HT29. But you can see here a majority of the tumor cells do not produce type 1 interferon. But we asked, would the supernatants from these interferon-producing tumor cells actually polarize a similar human monocyte 1-like cell? So we took human monocytes placed in culture and basically treated those with the tumor cell supernatants from those tumor cells that made type 1 interferon, the HT29 and NCIH1650, or two other cell lines that did not produce any type 1 interferon, the HCC827 or A549. And what we see is that these human monocytes treated with the supernatants that had interferon in them drove up the expression of these tumor monocyte 1 signature genes. These are all interferon-associated genes. That's pretty to be expected. But they were also able to drive up the expression of other pro-inflammatory genes like IL-6, TNF, CD80, CD86, etc. So it does look like human tumor cells that produce type 1 interferon are also capable of potentially polarizing human monocytes towards this monocyte 1-like state. So again, to pause and summarize where we are right now, we show that the MC38 tumors, we have more of these stimulatory monocyte 1 cells, and that is because the tumor cells themselves are making type 1 interferon, pumping that out in the tumor because of their active C-gas-sting pathway. So putting this all together, taking a bit of a step back, we want to know, is this whole phenomenon in any way associated with response to immunotherapy, like anti-PD-1 therapy? Is this tumor cell-derived interferon and associated monocyte 1 polarization, does it have anything to do with response to immunotherapy? So to start, we just looked at how these tumor models, the LLC and the MC38, respond to checkpoint blockade, anti-PD-1 therapy. And to maybe no surprise, these LLC tumors, where they're not making any interferon, there's more of these suppressive tumor monocyte 2 cells, you really don't see much of a response to PD-1 treatment. However, these MC38 tumors, where there's type 1 interferon, these stimulatory monocyte 1 cells, you see that there is this pretty nice response to anti-PD-1 therapy. So the question then became, is this response in any way associated with this tumor cell-derived interferon and monocyte 1 polarization? So to look at that, we basically took advantage of that TBK1 knockout MC38 tumor cell line that I mentioned before. So again, these MC38 TBK1 knockout cells do not produce any type 1 interferon, and I also showed they have less monocyte 1 cells, more immune suppressive myeloid cells. And compared to the ROSA control, which you can see actually treatment of PD-1 increases the survival rate up to about 40% of these mice being cured of their tumors. The TBK1 knockout tumors, they grow a lot faster actually, and the PD-1 treatment doesn't end up curing any of these mice. So it does look like this tumor cell-derived interferon and all of these downstream changes might be indeed associated with the overall response to checkpoint blockade. So adding that into the schematic here, we then wanted to ask the obvious next question. Okay, so we understand, what's going on with this tumor monocyte 1 polarization, but, we probably have in lots of patients tumors that are filled with these immune suppressive, potentially monocyte 2-like cells, where T cells are suppressed and you get resistance to PD-1 therapy. Can we somehow convert this suppressive tumor to look more like the stimulatory tumor, make an LLC tumor look like an MC38 tumor? And I'll show you how we got there. So to start, we wanted to say, well, why are these LLC cells not making any interferon, whereas these MC38 cells are pumping out lots of interferon? And what we found is pretty obvious, as shown here on these Western blots. You clearly see that LLC tumor cells completely lack the most upstream component of the cGAS-STING pathway, which is cGAS. So should there be any double-stranded DNA in these cells, it's not going to get detected, no interferon will be produced. Whereas MC38 has cGAS, STING, the whole rest of the pathway. But LLC actually has the rest of the pathway components present, just missing cGAS. So we asked, well, what if we just add back cGAS? So we made an LLC cell line that overexpresses cGAS, and you can see very clearly, you see activation of this whole pathway as indicated by phosphorylation of STING, phosphorylation of TBK1, IRF3, etc. And then these cells will actually start making some type 1 interferon. So we asked the question, well, if we take these LLC cells that are overexpressing cGAS, put them into mice, will they start looking like MC38? And will they become responsive to PD-1 treatment? And that's pretty much exactly what we see. So here, looking at the monocyte polarization, you can see that LLC tumors with the empty vector control transduction, you can see that they look pretty much like all the LLC tumors I showed you before, predominantly monocyte 2 cells. We've also shown that these, indeed are the more suppressive myeloid cells in this tumor. However, in the LLC tumors where cGAS is being overexpressed, the monocyte population looks completely different. It pretty much looks like MC38 tumors. So we have predominantly monocyte 1 cells and significantly fewer monocyte 2 cells as well. And what about the response to immunotherapy? Well, as I showed you before, the LLC control, the empty vector transduced control, no response to anti-PD-1 therapy. But when we overexpress cGAS, to start, we can see that those tumors actually start growing slower and you have an increase in survival. However, all of the mice eventually succumb to their tumors. But when we treat those LLC tumors overexpressing cGAS with anti-PD-1 therapy, we can actually cure some of those mice of their tumors. Now, it's only 30%, so we're not, we didn't cure cancer this time. But the point I want to make is this is a very aggressive and resistant tumor model. So even seeing some movement in the amounts that we can cure it's a high bar, and I think this is really encouraging. So with that, pausing and summarizing again, we found that LLC tumors not making any type 1 interferon because they basically have a break in their cGAS-STING pathway. We add back in cGAS, we can repolarize that tumor environment and make this resistant tumor more responsive to PD-1. So lastly, how does this all fit in with humans?
For cell lines that showed
the human ones. Yeah.
Did you look at the cGAS-STING pathway and
What I did is I showed that they produce interferon in a TBK1 dependent manner. So I used a TBK1 inhibitor, but that could be double-stranded RNA. It could be through a different pathway. So, but we didn't break down specifically and do the gene editing like it did in the mouse. But yeah, good question. Okay, so getting back to the relationship with humans. Well, the best way for us to really do this is by looking at clinical data sets. And most of those are bulk RNA sequencing data sets, and I'll show you how we projected some of our findings into those data sets. To start, we looked at
What happened if you would expose the LMC minus to just
yes, that works too. So we've taken out LLC monocytes and ex vivo, just add in some interferon, and they become yes, yeah. So what's interesting is that and I don't know if I think I took it out of the backup, but basically there's kind of a one direction of polarization that can happen. So you can take a monocyte 2 cell, these suppressive LLC cells, turn them into the stimulatory cells, and they do functionally change, but you couldn't really go backwards. So once they're the monocyte 1 cells, when we then basically had them in ex vivo and then took out that supernatant, put in LLC supernatant, they stayed as those stimulatory monocyte 1 cells. So there's some sort of it's kind of one direction there as far as that polarization.
And definitely when you receptors.
Yeah. Yeah.
What do they produce to you from that stage to M1?
Like
they must be producing something to the environment that is contributing.
the monocytes,
Yeah.
you're saying, yeah. So as far as like how they're actually having these functional differences,
Yes.
that's the kind of $1,000,000 question is, both from the suppressive side and the stimulatory side, whether it's a secreted factor, whether it's, something on their surface, and that there's a few things here and there in the literature, but as far as what's going on in our system, honestly, we don't really have a good idea. So as far as the human data set analysis. So we started by looking at colorectal cancer. And the reason for this, and this is TCGA data, and the reason for this is because there's different subsets of colorectal cancer, based off of their microsatellite instability. And microsatellite instability high patients, these MSI H subset of patients, are known to really highly respond to anti-PD-1 therapy or immunotherapy in general. And what's interesting is when we look at basically a gene signature that came from our mouse monocyte 1 cells and project that into this colorectal cancer data set, we see that these MSI high patients, these tumors have high expression and enrichment of that monocyte 1 signature. So that's one indication that this cell type is maybe associated with response to PD-1 therapy. Now, a bit more definitively, we looked at our own Regeneron clinical trial data from cutaneous squamous cell carcinoma in patients treated with our own PD-1 antibody, cemiplimab. And what we see is, we know from that trial who responded, who didn't respond. And what we see is that responders to cemiplimab had significantly higher enrichment and expression of those monocyte 1 signature genes as well. So all of that together is, a nice signal that what we saw in mouse, is potentially relating to what we see in humans, but of course, there's a lot more work to do there. And so with that, I'll summarize here. I just want to kind of take a bird's eye view and remind you where we started and where we ended up. We started by basically just trying to get a handle on the diversity on a molecular level of monocyte subsets in human and mouse tumors. And we found there's these different subsets, including this monocyte 1 cell type that we found to be immunostimulatory. And we were able to find a new way of identifying and marking these cells using 2 different surface markers that are unfortunately unique to mouse, but really helpful as we're trying to understand how these cells influence tumors in our models. And then subsequently, we really characterized how these subsets emerge. We showed that monocytes enter tumors and they pretty rapidly become differentiated and polarized. In an MC38 tumor, we see that the cells themselves are making type 1 interferons in a cGAS-STING dependent manner, driving this monocyte 1 polarization, T cell stimulation, and ultimately response to PD-1 therapy. Now, in an immune suppressive tumor like LLC, which didn't have any type 1 interferon production, we have these suppressive myeloid cells, and this tumor is resistant to immunotherapy, but we can simply target that cGAS-STING pathway and reprogram these monocyte 2 suppressive cells to become immunostimulatory and induce response to immunotherapy. And with that, this is an older slide because a lot of this work came from postdoc time, but there's a lot of people to thank and a lot of collaborations involved in this work from our immunology and inflammatory group at Regeneron. They and Demetris and Matt were all part of this bigger team. There's many members of the lab that contributed, but also we have a really powerful molecular profiling core that was responsible for a lot of the single cell RNA sequencing work. The flow cytometry group is really great at helping us sort out all these populations from tumors, et cetera. The mouse model generation and the postdoc program, which helps me along the way with this work. So with that, I can stop here and take any questions.
So going from non-zero to expressive non-two.
Yeah.
You're about 60 involved, or are there factors described cells that cause that?
There's definitely some factors coming from LLC cells, and I think it's shared. There's the same factors in MC38 cells as well. So when we get rid of the ability for the MC38 cells to produce the type 1 interferons in vitro and use that supernatant to polarize monocytes, they end up looking like monocyte 2 cells, and they're suppressive like that. So there's something they're making, because when we just leave them in bone marrow monocytes in culture, they don't just automatically become monocyte 2 cells. So there's, I think, growth factors and other factors that are coming from the tumor cells in both situations that drive that. But what that is, great question.
Do you have any desire to track that?
Oh yeah, I do. It's a high risk, high reward kind of project. So figuring out how and when, I think, will be key.
I have a question. So I'm thinking about the tumor cells and which ones express the S-thing and which ones don't express the S-thing. So do you think that it has to do with, like, well, two questions. One is, do you think that has to do with kind of the cell's origin that becomes a tumor? Is it kind of committed to CS staying based on that original state of the cell? But then the other thing I'm struck by is your CSCC data. The responders having higher MOD1 than the non-responders. And I would guess you would say that the responders are more likely to have higher CS thing than the non-responders.
Yeah.
And then if so, what is leading to that difference in the same tumor type?
Yeah.
Those differences?
So the first question about the cell origin and whether they're expressing CS or not. I mean, for the most part, every normal cell should express cGAS and STING in a certain way. Whether they have some sort of nucleic acid aberrantly in the cytoplasm that's driving the activation of the pathway, I think that's maybe more cancer specific. So I think because, like LLC, for example, when we put cGAS in, it turns on the pathway, there's something it's detecting. That was probably an advantageous tumorigenic evolutionary thing for the tumor cells to shut down that pathway and to evade the immune system. So that's my expectation there. For MC38, it actually is more of like it has a lot of, it's like an MSI high colorectal cancer model. So there's a lot of genomic instability, and that is known to drive up activation of the cGAS pathway. So that's probably why we're seeing it there. Just like how in the colorectal human data set in the MSI high, we see more of the monocyte 1 cells. So that kind of matches. Yeah, so I think for the cell type, it's whether or not there is, for some reason, DNA in the cytoplasm, it's driving the activation, or if they've just the cells have shut down the pathway. For the CSCC data set, we actually looked at the overall expression of cGAS and STING, and you actually don't see more cGAS and STING expression in the responders, if I remember correctly. So that wasn't really the most useful way of kind of differentiating those two populations. Yeah, for some reason it was just that signature did actually the best compared to a lot of other gene signatures that we tested. So I kind of think there would be something about those myeloid cells that really play a role.
So the kind of course of the whole organization, in vivo versus gene therapy, the publishing of DNA, are they comparable? Some neutral organization is much longer.
It seems relatively comparable. So the one thing that we really didn't get into today is monocytes eventually will differentiate and become macrophages. And everything that we see in the monocyte state basically just they keep going. You have these more MON1 like macrophages versus MON2 like macrophages, maybe M1, M2 like macrophages. But as far as the time course for the monocyte polarization, I would say the in vitro, in vivo are pretty similar. So here, and maybe didn't put all the details on the slide, but this is injecting monocytes into a tumor and then looking 2 days later. And this is what we see. In vitro, it's the same thing. We treat the monocytes in vitro and look 2 days later, and they look pretty much the same. So it does seem to maybe be capturing a similar, temporal dynamic. But what we're not showing, especially here, is what happens when you leave the tumors going. These monocytes are going to look more macrophage, express different markers that are more hallmark to differentiated macrophages. Cell line. So that's just the tumor cell line. Yeah.
You still see patients on before that.
Yeah.
So what else other than one?
Yeah. Good question. So there's two things I'll say to that. One is these were actually a pool of knockout cells. So they were not sorted, and it was probably about 80 to 90% knocked out based off of Western blot like kind of predictions. So there is still some tumor cell derived interferon coming from those non-knocked out cells. So that's one aspect. The other thing is, there could be so upstream of let's see if I have the pathway here. So upstream of TBK1 is the response to the double-stranded DNA, cGAS activation, production of cGAM. And what we've seen as well is that cGAM can actually leave the tumor cell. So it's also very possible that cGAM, and this has kind of been shown in the literature before too, and we thought was actually going to be the more likely suspect here, but cGAM can leave these tumor cells and enter in the myeloid cells or other cells that have a fully functional STING pathway in TBK1 and then produce their own interferon. So that could be happening. Yeah.
Do you think that this concept is tumors or it would be
Yeah. So for example, there's been, as far as like viral infections, there's been obviously a lot of papers about COVID recently, and there's definitely interferon associated monocytes in viral infections and shown to be immune stimulatory and secrete all these pro-inflammatory cytokines. So I think we're kind of recapitulating some of that in a tumor, right? Because this is an antiviral pathway that essentially is just active all the time, driving this kind of antiviral-like myeloid population. But as we were talking about before, like these tumor cells are also kind of making whatever's driving the suppressive phenotype. So it's kind of in between. It's a bit of a tug of war. And it's probably a bit more unique than what we see outside of them.
And then a different mechanism for the profile.
Yeah.
This M1 versus M2 would be found in any other tumor.
Yeah. We have to look. Yeah. And similarly, like how they would relate to monocytes and other tissues as well.
Would you advocate for interferon therapy?
Well, it's been done. I mean, that was one of the, I think, first approved therapies for like renal cell carcinoma, for example. And, we're still here. So I wouldn't, but I will say that I think where we in the field are really going is figuring out exactly which cell types really should be seeing the interferon, which cell types shouldn't, and making, because there are these reports about chronic interferon and driving resistance mechanisms. Obviously, these MC38 tumor cells, tumors they grow, and I think it's just because they've become accustomed to the interferon and are capable of growing in that context. So there's way more to figure out, I guess, in that sense. Yeah, but we are working out that.
Suppressive.
Yeah, that would be a great strategy as well. I mean, I think until we have a good, in humans, good set of markers, something that we can use to be specific, we're limited. But like you asked, figuring out how they become suppressive, how they acquire that phenotype, if we can figure that out, then, try to cut that out.
And it would be probably quite applicable to just a simple inflammation, simple MYM2 in atherosclerosis. And if you can figure out the switch